Everyone, let's go ahead and get started. We have a full slate today, and we're excited to present our arthritis series featuring rheumatoid arthritis. Thank you again for joining us. I'm Emily Boyd-Storman, the CEO of the ANRF, and along with our board of directors, staff, and those participating in the panel, I'd like to welcome you to the Spotlight series, the third out of a series of 5 featuring rheumatoid arthritis. We're so grateful for your participation and support and really appreciate your interest and time. Moderating the discussions today are 2 esteemed members of the ANRF Scientific Advisory Board, Dr. Peter Nigrovic and Dr. Elizabeth Mellons. Dr. Nigrovic is a professor of pediatrics at Harvard Medical School and is also the chief of the Division of Immunology at Boston Children's Hospital. Dr. Mellons is a professor in the Department of Pediatrics, Divisions of Rheumatology, Immunology, and Human Gene Therapy at Stanford University. Before we start the webinar, we have 2 logistical pieces of information for you to note. First, this webinar is being recorded and we'll have it available on our website at a later date. Second, if you have questions throughout the presentations, please type them into the Q&A and we'll try to get to all of them. If we aren't able to get to yours, please email info@curearthritis.org after the webinar with your question. I also want to mention that we have 3 presenters with us today, as Dr. Shuri is unfortunately not feeling well and unable to present. Now, let's move on to the presentation featuring the impressive work of our ANRF scholars. Without further ado, I'm happy to turn it over to today's moderators.
Well, thank you. Thank you for that lovely introduction. My name is Betsy Mellons, and I am currently, as Emily mentioned, I'm currently a professor of pediatrics at Stanford. I was trained in pediatric rheumatology, but the focus of my work really for the last large number of years has been laboratory-based investigation into immune mechanisms that are relevant for rheumatic diseases. And I've had a particular interest, although not exclusive interest, in some of the diseases that affect children. I'm also really focused on mentoring of the next generation of investigators, which is part of what makes this, this event today particularly exciting. My work was really, there was a real important addition to the building of my program by my getting an ANRF grant at an early stage. And I really want to thank the foundation for that. And I really love the fact that I can give back as a member of the Scientific Advisory Board, participate in events like this, and also be able to stay in touch with what the newest up and coming researchers across the rheumatic diseases are doing. It's a great opportunity. Plus the rest of the Scientific Advisory Board is really fun to work with. So with that, I'll turn it over to Peter to introduce himself, and then he'll introduce the first speaker.
Thanks, Betsy, so much. So I'm an adult and pediatric rheumatologist at the Brigham Women's Hospital to start my career, and then currently Boston Children's Hospital, where I'm the chief of immunology, a division that includes rheumatology among other groups. So I'm also a lab investigator whose transition from postdoc to principal investigator was entirely facilitated by the ANRF when I got an ANRF grant in 2009 on a project that is now a renewed R01 in my lab, sort of a major grant that supports a major aspect of our work. So that work has had legs, I would say, and we have been privileged to also mentor a number of ANRF fellows for whom it's also been a critical juncture of their career. So I'm delighted to participate here and as a member of the Scientific Advisory Board. So we'd like to move on to the science and to hear from the really kind of exciting work that the ANRF sponsors. As listeners may or may not appreciate, the ANRF is a specialist organization. It funds particularly extremely promising young investigators at the juncture where their career's just taking off. That's exactly where I was funded, and that's where our current investigators are. So I'll start with introducing Dr. Helena Jonsson, who is one of my colleagues at the Brigham Women's Hospital and actually co-directs with me a clinic there called the Center for Adults with Pediatric Rheumatic Illness. She's an MD, Ph.D. investigator and a faculty member. and has been doing really exciting single cell work on rheumatoid arthritis tissues and fluids. So I will look forward to hearing that work. Helena, please dive in.
All right, thank you so much. So let's see if I can get this presentation up and running. All right, hopefully that looks good. All right, so thank you so much for this opportunity, and also thank you for the ANRF funding that I received in 2018 that helped to really get this project going. Another project with legs. It's still in the much earlier phases than Dr. Nigrovic's project, obviously, but such an exciting study that I'm happy to share with you today. And so I study a cell type called Granzyme K positive CD8 T cells, and that's that we've found that drives joint inflammation in rheumatoid arthritis. So I don't think I need to give much of an introduction to rheumatoid arthritis to this particular audience. I think everyone else already knows, but rheumatoid arthritis can be, a really destructive disease. And even, in 2021, I just had clinic this morning, and I still have patients who are struggling to get the joint inflammation under control to try to avoid having hands that look like this, having knees that are destroyed, et cetera, et cetera. And so this is still, a big deal illness that we're dealing with, and we're always looking for new mechanisms, new targets for treatments, things that we can give to patients to help them feel better and to keep their joints working well. Now, what is joint inflammation? So joints are not really an organ that's talked about, in biology classes, even in high school, et cetera, et cetera. I think I was probably a rheumatology fellow, a rheumatologist in training before I ever saw a picture of what a normal joint tissue should look like. So in our hands and in our knees and everywhere like that, where two bones come together and bend, there's a very thin joint lining that's called the synovium. That's sort of the technical term. And if you take a tiny little piece of synovium out of a normal person's joint and look at it under the microscope, it looks like this picture here on the left. And what you see here is sort of a light pink wash with a little bit of a purple layer up here. And this pink here, this is collagen. This is connective tissue that makes this tissue strong, but also flexible. And it's kind of like, you can think about it kind of like a balloon, strong, but very flexible and very sort of, moves along with you and doesn't impair the way that you use your hands or your knees or other joints. And it just has a few cells up here at the top that have some functions that have to do with lubricating the joint and things like that. Now, if you take a little piece of synovium from somebody with rheumatoid arthritis or, psoriatic arthritis or juvenile idiopathic arthritis, pretty much any kind of autoimmune arthritis, it will look something like this image here on the left. It's enormous, and you can see it's a completely different color from this image here on the left. It's full of these tiny little black dots. Like if you get up close to the screen, you'll see there are thousands and thousands of little black dots here. Each one of those is a cell. Most of those cells belong to cells in the immune system, because the immune system has traveled to this joint capsule, to this joint tissue, to start attacking something that we don't yet understand what it is. For some reason, the immune system has gotten really angry and has gone into this tissue and just caused all kinds of havoc. And one of the big questions that rheumatologists and that immunologists and scientists have been looking for the past 30 or 40 years is, what are these cells? Like, what cells are present here? What are they doing? How are they interacting? Because those are the questions we have to ask in order to start asking the next series of questions, which is, how do we get rid of them? How do we break those interactions? How do we turn a tissue that looks like this into a tissue that looks like that? And so early on, people have been finding cell types here sort of one by one. And this is a cartoon image taken from a review written about 10 years ago now. I'm sorry I didn't provide the citation here, but there are different cells in the immune system, and T cells and B cells and so forth. And we had some idea about how they work together, but this was taken from a review written for scientists. And you can tell that there's not a lot of detail here. I mean, it kind of looks like something you'd find in the Sunday cartoons in the paper. And so we really have a ton left to learn here. And that's something that's happening right now. And so this project was born in exactly that kind of study, the kind of study that's trying to provide much more detail than this cartoon on the left. And that's this image here shown on the right. So the image shown on the right was produced using a new technology called single cell RNA sequencing. So single cell RNA sequencing means that we take a tissue. In this case, we take synovial tissue biopsies from people with rheumatoid arthritis. So tiny little tissues, and we digest them so that they're sort of a solution of one cell all by itself at a time in, tiny little tubes. And we send these cells through a machine that collect all the mRNA in the cells. The mRNA, these are the messages of the things that the cells produce. Each cell, they produce various proteins and other kinds of things. And looking at those kinds of proteins can tell us about their jobs, about how they're viewing the world and how they're interacting with other cells and things like that. And so by looking sort of at this mRNA, we get a much more detailed picture than we had before. I know it can, in some ways, like I made this slide and then I was like, wow, this kind of looks like art from the 18th century, where like everything looks like exactly what it's supposed to be. And then the image on the right looks like a Jackson Pollock painting or like a Picasso or something like that, very abstract, but it contains much more information that's really been revolutionary. So where was my project born? So if we zoom in on one particular kind of cell in the immune system, that's a cell called the T cell. Now, we already knew that there were T cells in the synovium, but there are two kinds of T cell, and all of the attention had gone to one. But here in this study, we looked at both kinds of T cells at the same time, and we asked, what kinds of T cells do we see there? And here, what you're looking at is, again, the single cell RNA sequencing data. Each tiny little dot that you see here is one particular cell, and it's put on a map based on like what kinds of things it expresses. So you can kind of say that it's being sorted by the kind of cell that it is. And then we've categorized them here into 5 different categories. 3 categories in shades of pink. These are CD4 T cells, which are one kind of T cell. And then we have a category in green and a category in blue. Now, as I mentioned before, CD4 T cells, it's been known for 20 or 30 years that they're really important in rheumatoid arthritis. And they kind of steal the spotlight, if you ask me. When people think about T cells in rheumatoid arthritis, they almost always think about CD4 T cells. And they represent about half of the T cells in rheumatoid arthritis joint tissue that's inflamed. But what about these other CD8T cells? Like, they're almost as numerous, and they're probably doing something there too, right? What role do they play? Nobody had really studied this in much depth before, and I think that there's a pretty good reason for that. And to think about why these cells had been dismissed before, let's think about what CD8T cells do. So many of you have probably never heard of CD8T cells before, and that's completely understandable. CD8T cells are something we take for granted, maybe until this COVID-19 pandemic, to be honest. So the traditional role for CD8T cells is that they are the cell that sort of scans your body for virally infected cells. So cells that have the COVID-19 virus, for example, or a cold virus, or, like a cold sore on your mouth or something like that. Those are the kinds of cells that they're searching out for. And what they do when they find them is they release this package of chemicals, these proteins that called Granzyme B, perforin, granulysin. These chemicals here, they attack the cell that's infected with the virus and they kill it. And so these cells are often called cytotoxic T lymphocytes, cytotoxic meaning killer. And so you can think of these cells as being sort of the cell's assassins, and they're just sort of scanning the body for virally infected cells, for cancer cells, and killing them. And that makes a lot of sense. They're very important for viral infections. But what role could they possibly be playing in rheumatoid arthritis? It wasn't really clear, so no one really dug into that. But this is actually where the crux of the matter comes in, because it turns out that those killer cells, those are only a small portion of the CD8T cells we find in synovium. Only this tiny little portion down here in blue have the genes like Granzyme B, like perforin, like granulysin, like those chemicals I told you that are released towards target cells. That's only a tiny portion of the CD8T cells in rheumatoid arthritis. It turns out that the big cluster of cells here, they're totally different. They don't express these proteins that kill other cells. They must have some other role. And this is a cell type that until this study had never been observed before, a cell type in rheumatoid arthritis synovium where we didn't know, what they were doing, where they came from, how they developed. They expressed this protein called Granzyme K that was essentially also a mystery. Only a couple of other scientists had ever studied it, Granzyme K, before. And so that's another thing that we don't understand very well. So here, in this disease, rheumatoid arthritis, that we're always looking for new treatment targets, we found an entirely new population and an entirely new protein target with Granzyme K. So what does Granzyme K do? So I'm not going to go through all these, like fancy studies and things like that we've done. I've sort of summarized them all here in this little cartoon of my own. But it turns out that these Granzyme K positive CD8 T cells, they don't kill other cells, but they do incite inflammation. And they do it through multiple mechanisms, all right? So first of all, what does Granzyme K do? So Granzyme K itself produces inflammation. We found that it interacts with things on the outsides of cells. And so what I'm showing here is a T cell that expresses Granzyme K here in green. This pink cell here at the bottom, that's any of the other synovial cells that you saw in that really inflamed synovium. We've looked especially at fibroblasts, which are sort of one of the network cells in joint tissue or in synovial tissue. And what we found is that Granzyme K can interact with proteins on the outside of these cells and lead to production of cytokines like IL-6 and chemokines like CCL2. IL-6 is one of the really inflammatory cytokines in rheumatoid arthritis. This is a cytokine that's been targeted with treatments like tocilizumab, also known as Actemra, and sarilumab, also known as Kevzara. This is something that we know is really important in rheumatoid arthritis. We also see chemokines like CCL2. A chemokine is a kind of chemical that attracts other immune cells to come to the joint. And so this is also attracting more inflammation and more inflammatory cells. We've also found that Granzyme K can enter the insides of the cells and act on other parts of the cell, such as the mitochondria shown here. And that releases something called ROS, or reactive oxygen species. This is a special kind of oxygen molecule that also acts in pro-inflammatory ways and that we know plays important roles in the inflammation that we see in rheumatoid arthritis. So, so we have these new mechanisms of Granzyme K mediated inflammation. Granzyme K positive CD8 T cells also make TNF and interferon gamma. These are two cytokines or immune system communication molecules that also react with cells to produce inflammation. And what we found is that interferon gamma and Granzyme K, they act together. So each one has a partial effect, but if they're both present, then they work even stronger. So they add on top of each other and have an even bigger effect. And so what we believe happens is that Granzyme K positive CD8 T cells, we find them in the blood at low levels. Those are the cells that enter the synovial tissue, and in the synovial tissue, they're activated, they proliferate, and they release Granzyme K, interferon gamma, TNF, and these molecules act on cells in the synovium, such as fibroblasts, which then in turn release other inflammatory molecules. And this is part of the reaction that causes the snowball of inflammation in rheumatoid arthritis. We also think that these Granzyme K positive CD8 T cells return to the circulation. So they return to the bloodstream and then can travel around the body and go to other joints. And that might be one of the ways in which the inflammation is sort of acting at multiple different joints at once. And so this is, again, a work in progress. And so some of these things we've found hints for, but we need to confirm, especially, the sort of migration into and out of blood vessels. But we think that this cell type could be one of the keys to sort of driving inflammation in rheumatoid arthritis. And so this project has been a teamwork effort by multiple different individuals. I am an instructor in the lab of Michael Brenner at Brigham and Women's, and I work with a technician named Emma Gomez-Rivas, who's done a lot of the work that's gone into this project, and several other members in the lab have helped. A lot of the fancy RNA-seq analysis was done as a collaboration with Fan Zhang, who's a very talented scientist in the lab of Shoma Rachowdhury. She'll be moving to the University of Colorado soon, so I'm very sad about that, but happy for her. And then Deepak Rao and Garrett Dunlap are two other individuals here at the Brigham who've helped out with this. And again, this couldn't have been possible without funding from the Arthritis National Research Foundation. So thank you very much for that.
Helena, that was wonderful. Thanks for the summary. I think that I see a question in the Q&A, but that's not directly related to this one. We can maybe address that later if that's what Emily prefers to do.
Sure.
But should we ask a few questions about this presentation first?
Absolutely.
That'd be best. So I've seen some of this work evolving over time, and I think it's fantastic and important and a really neglected cell in this disease. Do you have a sense for what the pathways are that recruit these cells and retain them in the joints?
So we think that they're recruited by different chemokines. So chemokines are a name for sort of a class of molecules that attract other cells. And it turns out that these Granzyme K positive CD8 T cells, they express receptors for a certain group of chemokines, but not other ones. And so the cytotoxic T cells, they express, they're attracted by some chemokines, and the Granzyme K CD8 T cells are attracted by other chemokines. And so we think that there's a difference there that allows the Granzyme K positive CD8 T cells to travel to the joint more frequently than these Granzyme B positive CD8 T cells. They also seem to stay for some reason, whereas the Granzyme B positive CD8 T cells, the ones that sort of smaller blue cluster, the Granzyme B cells seem to tend to leave, and we haven't figured out like what that stay leave signal is. That's something we're still working on.
Do they are they activated in an antigen specific way? That is, are they recognizing a specific target, or are they just responding to the inflammatory environment?
We have conflicting data on that. Like, initially we thought that they were antigen independent, meaning that they were receiving signals from, inflammatory cytokines produced by other cells and so forth, and they weren't seeing like a specific antigen. But actually newer data that we have suggests that they might actually be antigen specific and acting that way. So that's new TCR repertoire data where we look at and we see some what looks like antigen dependent expansion of particular populations of this. And so that's still an area under investigation.
So I have a question, but I also see there's a question in the chat. I think I'll ask the question in the chat first from Krista Horn, and then I'll ask my own. So the question is, can blood work be taken to find out how much of these cells are in the body?
Yeah, so the funny thing is that these cells are very rare in blood. Even in people with rheumatoid arthritis, they're no more common than in people who do not have rheumatoid arthritis. And so we haven't yet figured out a blood test for this. Like, we have to look in the tissues. Interestingly, people with rheumatoid arthritis seem to have that cytotoxic B cell. They have higher levels of that, or that type of CD8 T cell, not the sort of the Granzyme K one that we're talking about. So there are still a lot of like mysteries here about how this is working and what regulates the levels of these different cell types.
So my own question has to do with other types of arthritis that are inflammatory types of arthritis. I realize that your focus is RA, and doing single cell on synovium is not a snap, but I just wonder whether, with your having found these, other people or you have looked in other settings of inflammatory arthritis to see if they're there also.
So yes, we've looked at psoriatic arthritis, we've looked at juvenile idiopathic arthritis, and the cells are there also. So it seems to be a cell type that's present in many types of inflammatory arthritis. We've also actually found it in bowel from people with Crohn's disease and ulcerative colitis. And so we think that, and even in kidneys that are affected by lupus nephritis. And so we think that this is actually a tissue CD8 T cell type that's present in many different contexts, even COVID-19, like even infections, the cell is present. And so, we're really curious to see like what, where this project goes, because we think it will shed light not only on rheumatoid arthritis, but also on a number of other diseases.
Yeah, that's great. That is great. Okay.
We move on. I think we want to make sure we have time for all.
Yeah. Okay. So our next speaker is Dr. Tiphanie Vogel. And Dr. Vogel received her ANF Scholar Award in 2017. She is an MD Ph.D. researcher, and she is currently an assistant professor of both pediatrics and medicine at Baylor College of Medicine. And she's going to continue the theme in terms of talking about inflammatory arthritis, although I think we'll hear from a pretty different angle.
let me share. You see my screen?
Yes.
Perfect. So thank you for inviting me. I feel very honored to be able to share some of our research today. Similar story to the other investigators that you heard, our work would not have been able to be launched without the support of the ANRF, and it meant a lot to my career. So I'm going to start by explaining why it is that we use rare disease models to help understand more common immune disease. The reason being, you know, sometimes I feel like a little bit like I have to justify why we study something that maybe like less than 200 people have, so that we can understand the immune system better to help people who have more common immune problems. And then I'm going to show you a summary of some of the work that we've done that tries to link the findings that we have from studying rare disease with potential movement on therapeutics for inflammatory arthritis. So one of the most common questions that I think rheumatologists get when someone's been diagnosed with a rheumatologic illness is like, why did this happen to me? And, you know, I often wonder that about my patients as well, because obviously most people don't have swelling, stiffness, and pain in their joints. So why is it that some people do? And so, you know, physician scientists like myself and my colleagues that are presenting today, this is one of the things that we're trying to figure out over the course of our careers and with the science that we do, because we are under the impression that if we figure that out, that we may be able to prevent or at least treat these inflammatory problems. And so, you know, the immune system is very complicated. Studying it, all of the moving parts of it at one time is a little bit too much for anybody, even with super impressive single cell like you just saw. And so there's other ways to simplify studying the immune system, and the way that we take, the approach that we take, is to study rare disease and narrow down what we're looking at by focusing like a magnifying glass on these rare problems and then extrapolating what we learn to more common problems. And this isn't like a new technique. So I really love this quote because I consider myself a rare disease doctor, more than 400 years old, talking about there's no better way to advance medicine than by to really look closely at rare disease. And so this is of course our approach. So the ANRF award allowed us to more closely study a very rare syndrome called STAT3 gain-of-function syndrome. I'm going to describe it to you briefly, but there's less than 200 people that have this in the entire world. So if you're like, I've never heard of this, trust me, neither has your doctor or pretty much anybody else you know. So we were able to recognize about 7 years ago that there was a small collection of patients who had significant burden of autoimmunity. And of course, these patients tend to be children, but this can present in adulthood as well. And the major feature is that these patients have multiple autoimmune problems at the same time. And that includes not just the joints, although 20% of the patients with this problem do have a very inflammatory arthritis, so nearly indistinguishable from rheumatoid arthritis or juvenile arthritis if they're children. But they can have autoimmune problems of the thyroid, the lungs, the intestines, the skin. Their pancreas can have an autoimmune problem, which leads to diabetes. And in addition, other major features of this problem are not quite understood why this happens, but the patients are often very small or short in stature. They have lymphoproliferation, which is describing a swelling of the lymph nodes, spleen, and liver. And their immune cells are being destroyed by their immune system. And so they have autoimmune cytopenias like low blood count or white blood cells or platelets because of their immune disorder. In addition, quite a number of the patients that have this problem, their subset of immune cells called B cells that make immunoglobulin to protect you from infection, they don't work right. And so they actually have low immunoglobulin levels and are at risk of infection. And so, you know, this very rare disorder is a significant disease burden for these patients. And we think that if we dissect out what's going on with this condition, it will help us understand what's going on with more common inflammatory arthritis. And so, so why does this happen to these patients? Why do patients with STAT3 gain-of-function get so much autoimmunity. And in order to understand that, you need to know, of course, what STAT3 does. So as we just heard about, cytokines are part of how the immune system talks to each other. And when cytokines, which is represented here by IL-6 with this little pink ball, are able to interact with their receptors on cells, their signal is transmitted into the cell. And almost all cytokines, although not all, signal through a certain very classic pathway that immunologists study called JAK-STAT signaling. And so the activation of the receptor by the cytokine activates the JAK molecule, which then propagates the signal down into other STAT molecules like STAT3. And when STAT3 becomes activated, it works as what's called a transcription factor, which is a protein that goes to the nucleus and binds DNA and tells other proteins to either be made or stop being made. And so we are well aware for a long time that this is a very important pathway in the immune system, and that activation of STAT3 is like critical to multiple immune processes. And in fact, this may start to sound more familiar because a lot of patients take medication that blocks aspects of this pathway to control their inflammatory arthritis. So we can block IL-6, just like you heard. We can also use JAK inhibitors to block intracellularly this type of cytokine signaling. So what we were able to show when we discovered this disorder is that the patients who have STAT3 gain-of-function syndrome is named that way because they have an overactive STAT3 molecule. And this is a schematic where the STAT3 protein's been like stretched out, and you can see each of the individual mutations in patients that have been reported. And as we were collecting this cohort and curating this information, we realized that like quite a number of the patients that had mutations in what's called the DNA binding domain had arthritis. So only 20% of all of these patients have arthritis, but tends to be clustered in these patients that have DNA binding domain mutations. And so we wanted to look at that further. And what we decided to do next was to literally measure the DNA binding of the different STAT3 molecules. So this is still my kind of like schematic of cytokine signaling. The pink arrow is pointing to the area that we're testing to report this data. So we're literally utilized a tool called ELISA, DNA binding ELISA, to measure how strongly these different STAT molecules bound to DNA. And that is summarized in this graph. So if the DNA is bound tightly by the STAT3 molecule, this bar will be higher. And so you see here in the red that with stimulation or cytokine signaling being implemented in these cells that are derived from either STAT3 gain-of-function patients, these different mutations, or a person who does not have STAT3 gain-of-function and therefore have wild-type STAT3, that there's significantly higher DNA binding in the patients with STAT3 gain-of-function. So our idea that something was going on at that location with these molecules, we feel that this supports that idea. And so then we were thinking like, well, if their STAT3 is overactive and it's because it's binding DNA tighter, is there a way we can like interrupt that DNA binding and like thereby reduce the inflammation? It seems to just follow from what we were like our original hypothesis was. So then the next step we did is we kind of like tried to look at that. So as I showed you, you can block at the IL-6 receptor, you can block at the JAK level already with therapeutics that are available. But like maybe what we need is another therapeutic that can block STAT3 itself. And those are in development, but they are not yet available. So what we did was we took a variety of STAT3 inhibitors and we tested them and looked to see if they could inhibit wild type STAT3 or STAT3 gain-of-function. So in this assay, we're looking where this pink arrow is, we're looking at downstream after STAT3 is turned on, is there overactivation of the downstream pathways, and one of those is called SOX3. So these bars are measurements of SOX3 that are done by real-time PCR. And you can see that when we stimulate cells that are derived from either someone who doesn't have STAT3 gain-of-function or is wild-type STAT3, or patients with these different STAT3 gain-of-function mutations, that we do stimulate using the cytokine STAT3 activity, and that we can, using a STAT3 inhibitor called S31201, reduce the amount of activation of STAT3. And in some instances, it's only a small amount, such as with this mutation here. In some instances, it's quite a significant amount, like half of the activity of STAT3. We used tofacitinib, which is a JAK inhibitor, as our control. And so you see that's a very effective and good positive control because all of that signaling is inhibited. So this is kind of suggesting that there could be the possibility of modulating STAT3 activation using STAT3 inhibitors that interfere with DNA binding, as that's the predictive mechanism of this S31201. And then the last piece of data that I'll show you is that, we're committed to trying to advance these therapeutics as a new option. We've developed a preclinical model to look at this in, which is like essentially a mouse model of STAT3 gain of function. But in the meantime, I have lots of patients that need therapy, and we can't wait for all the next therapeutics while we're developing them. We have to treat our patients. And so we had this idea that perhaps targeting this IL-6, JAK, STAT3 pathway would be more effective for a certain subset of patients than for other patients. That also makes sense. Not every medicine works well exactly the same for every patient. And so could we look in the blood of patients with arthritis? And in this instance, we looked at patients with poly-JIA, which is at diagnosis, they have at least 5 or more joints affected. And we believe that that's the most like rheumatoid arthritis of the juvenile arthritis subtypes. And so we looked at activated STAT3. So in this, again, the schema, the pink arrow is now pointing at this phosphorylation site on STAT3 as a surrogate of STAT3 activation. And then these dots are representing how much STAT3 activation or phosphorylation has occurred in the blood of the patients when we take their blood out and look at it. So compared to adult healthy controls, you see that children who have poly-JIA have higher amounts of activated STAT3 when they're diagnosed with their arthritis, and that a subset of these children really have quite higher amounts of activated STAT3 than the average adult. And this level goes down when we treat them. But these patients were actually all treated with a different type of cytokine blocker and not anything that's targeting the IL-6, JAK, STAT3 pathway. So what we're looking to do in the future is actually pull this high subset of activated STAT3 patients and see if we can get better treatment outcomes if we treat them with a tailored approach. And so just in conclusion, hopefully you've convinced you that rare disease models can help us better understand complex immune disease, because that's what we do. And that, of course, STAT3 is a critical molecule. Everybody already knows that in human immunity, but that if we tailor its activation, we can maybe reduce inflammation. And we are looking at doing that in a targeted or tailored manner to help our patients with better outcomes. And I'd be happy to take any questions. This is my lab. My very first grant was an ANRF grant. So I have been able to go on from there, and I'm happy to keep doing so in the future. Someday I want to be like you guys, and that's my goal. But thanks for having me. Let me stop sharing.
Okay, first, Emily, are there any questions in the chat, or shall I go ahead with my questions?
Go ahead with your questions, Dr. Mellons.
Okay, so first I just want to start with sort of a big type question. So 200 children around the world with gain of function STAT3, how do you find them? And how did you know how did you even find, how was the first one found that might have led to a search for the others?
I love this question. I had to cut some of the good stuff out because you guys said only talk for 10 minutes, which I probably went over anyway, but okay. So this is an excellent question. I focus on extreme phenotypes, and these patients are not subtle. So you probably have had someone in your clinic where you're just like, whoa, what is going on? And in the old days, which is literally like 10 years ago, we really couldn't do much about that. But because the geneticists, which I'm not a geneticist, but I'm really thankful for those guys, they made genetic sequencing so much easier that you really can like take patients with extreme phenotypes and ask if they have something like STAT3 gain of function. So that's how, the original patient was just a patient that when I met him on my second day of fellowship, I was like, whoa, what is going on? We have to figure out what is wrong with this child. And after you do figure that out and people know that these disorders exist, they can test for them much easier now. And so there probably are more than 200, and there's probably, you know, just sections of the world that don't have such access to genetics like we do in the US or other places. But slowly, I think, you know, now that this is a known disorder, when people have the ability to look for it, that's how we find them. And then how do I get them? I mean, I think I'm like known to be like a weird person that wants to know about STAT3 gain of function and get emailed when people discover these patients so that we could perhaps collect samples from them to advance our work. So if anybody knows anybody with one, we would be happy to help them with that patient.
Can I ask a question about the broader application of this? So I thought your data on the phospho-STAT3 was really interesting and how there's that broad range. So this was whole blood or was this a subset of cells?
So, you know, we bank them and run them together. So those are banked peripheral blood cells that are then, you know, reanimated and tested. I showed you data with activation by IL-6, but we've also checked with IL-27 and interferon alpha.
And do you know is it a particular cell subtype that exhibits this phospho-STAT3 or is it everybody?
When I want the histograms to look really good, I gate on the CD4s. But yes, I showed you the data from the total, but it's mainly driven by CD4 T cells. All CD4 T cells will activate STAT3 when you put cytokines on the peripheral blood. Only a subset of CD8T cells do. So I actually don't know that anyone knows why that is, but that we think it's being driven by the CD4s. We know that IL-6 signaling is really important in the development of different CD4 T cell subsets. So.
Dr. Mellons, we got a question in the chat if you want to read that one. I think you're on mute.
Yeah, okay. So would this be reasonable to ask for evaluating STAT3 gain of function mutation in RA patients in the future? Should that be looked for?
This is a great question. I'm going to answer it, but then I'm also going to change it to like what I think is an even better question. So I will tell you what I tell people to look for STAT3 gain of function. If the patient has arthritis and another solid organ autoimmune problem, and by that I mean like also inflammation of the intestines or also inflammation of the pancreas leading to that type of diabetes or also lung disease, and they are either short, very short, or they have autoimmune cytopenias, like their blood counts are low because of the immune system, then you should check them for STAT3 gain of function. So that's like kind of my rule. But really what I want the question to be is, should we look for highly activated STAT3 in patients when they're diagnosed? And then should we tailor their therapy, not start with a TNF inhibitor or something else, but start with an IL-6 blocker or a JAK inhibitor? Yes, like we can't quite do that yet, but hopefully that's where we're headed so that we can like profile with conventional tools, nothing super fancy that can't be done quick enough. And then ID those subset of patients that might, you know, not have to trial and error until they arrive at the idea that this is the right medicine for them. So that's kind of our idea or our like direction we're going.
Thank you, Tiphanie. I think
you're welcome.
I think how short is short? Just quickly answering the question. One last question popped up before I move on.
Yes, when these patients are short, they are more than 2 standard deviations from what they should be for their age or for the adult height. So like less than 5
Okay.
Mm-hmm.
Okay, there's one more question. Maybe you can answer it in the Q&A. And then if we could move on to Dr. Theresa Wampler Muskardin. So she is also adult and pediatric trained. There are 3 of us here on this panel. That's rather unusual to have so many at once. There aren't that many of us. But she is an assistant professor at NYU and has done a lot of work in lupus. And this work, however, is on rheumatoid arthritis and interferon signaling. So Theresa, please dive in. You were funded in 2019 by us. So excited by that.
Yeah, more recently. So it's been fantastic, and much like you've already heard from the panelists and other presenters. It's been funding at a critical time in my career and has allowed me to really dive in and launch in an exciting direction that just lays groundwork for additional studies and future work that I'll at least give you a glimpse of in the next 10 minutes. So let's see, share screen.
Just to guess, Tiphanie, did you stop sharing? You must have. Okay.
Yes.
You did.
And I put share screen.
You're sharing from the bottom?
Yes.
Like, okay. And are you seeing a set of choices to share?
Yeah. And I clicked desktop and share again. Grant access.
Do you want to send a PowerPoint really quickly?
Sure. Sure.
And send
Let me
it.
go ahead and send it to me, Theresa, and then I'll send you my email in the chat. And then if you want to just go ahead and get started, and we can walk it through while we move it.
Okay. I just sent it.
So you could probably just start to give a little bit of a brief overview of what you're going to talk about before the slides get there.
Okay. So my work is in precision medicine and rheumatoid arthritis. And Peter is exactly right that as a fellow very early on before, with my first research mentor, I did study type 1 interferon pathway which is most known for studies in lupus. But I've always been interested in inflammatory arthritis, and even in the earliest stages, really was trying to investigate it as sort of an immune phenotype to help us understand differences between patients with heterogeneous disease or more complex disease. So my current line of work that has focused in particular on rheumatoid arthritis really has allowed me to break new ground. So I do work in a lab that has particular expertise in the type 1 interferon pathway, which I'll talk about very briefly when the slides are up. It's shown to have some great impact in treatment response in rheumatoid arthritis. Okay. So do you see presenter view, or do I only see presenter view?
We see presenter view at the moment also.
Yeah.
Emily, do you want to just put it onto regular view with the slide showing as the whole field? And then Theresa can just say next slide.
Yes.
Mm-hmm.
I can figure out how to do that. Sorry, everyone.
My apologies too. I have
Wonder if it's
shared
under display settings at the top.
Okay, that's good. That's good.
Great. So Helena did a fantastic job with background information, and of course, many of you also don't need in-depth background information with regard to rheumatoid arthritis. So thank you, Helena, for your fantastic presentation. It's a very common systemic inflammatory disease, but characterized by a destructive arthritis and accelerated atherosclerosis. And we have many treatment options, and biologic therapies in particular have allowed remission to become a realistic goal, to become our treatment goal for patients, but we have no guidance on which therapy to use in a particular patient. And yet we know that remission at 3 months is the best predictor of remission at 1 year. But our trials of treatment, which is our current strategy, it's really trial and error because we have no guidance on which therapy is best for a particular patient. A trial of a treatment takes typically around 3 months, and yet we want remission at 3 months. So we're really behind the clock, and I think it's imperative that we better understand patient subgroups so we can identify what treatment is best for a particular patient, similar to what Tiphanie was talking about with STAT3. So it also mentioned we're TNF inhibitors, and that's because TNF inhibitors are the most common initial biologic employed for treatment of inflammatory arthritis and rheumatoid arthritis in particular. But much like our other therapies, even for TNF inhibitors, which has allowed remission to be possible for some patients, there's a heterogeneity, there are differences in response, and a third of patients have no response, another third have only partial response. So after a trial of therapy, there are about 60% of patients that have some level of disease activity, and we're aiming for remission or really low disease activity. Oh, next slide. So my work in particular has focused on predicting treatment response to TNF inhibitors as a good starting point, given their commonality in being prescribed, and they're expensive and they're not exactly benign if given to someone who might have an adverse reaction. So again, with the aim to get the right medicine to the right patient at the right time. So a little very brief, concise thumbnail about type 1 interferon. It's a cytokine, and as the previous presenters explained, cytokines are really messenger molecules that our body uses to communicate. And I think the definition on the top is the most concise, but also the most accurate definition of type 1 interferon as a cytokine. It's really a pleiotropic biologic response modifier. And really what that means is it impacts every cell type it touches, whether it's an immune cell or not immune cell. It's that powerful, and it's powerful at very small amounts. It also induces a broad spectrum of biologic activities. Interferons are named for their ability to interfere with viral infection. And for that, we know they have a key role in the initial, what we call the innate immune response in all cells. But at the very same time, they impact and influence the adaptive immune response. And they enhance specific antibody production, which is our secondary, slower process response. I want to mention, because it's relevant for the rest of the talk and my work, that in humans, that there are 2 major type 1 interferons studied and relevant, and that's interferon alpha and interferon beta. And we know a lot in autoimmune disease with regard to alpha, and in particular in lupus, because patients who have high interferon in lupus, the majority is interferon alpha. But interferon beta is also very important, whereas alpha is known for its role in enhancing that adaptive immunity, Both are known for roles in antiviral immunity. Interferon beta is particularly important for priming cells for subsequent responses. So recall Tiphanie showed that in one of the models, actually in Helena, when there's additional context, like other cytokines hitting the cell, sorry, this is from Helena's model, like TNF and interferon gamma, it's impacting the effect of other cytokines hitting the cell. So it's not as simple as one surface molecule and then downstream effects necessarily. And interferons work in this way. They really impact how the cell responds to subsequent stimuli in the context. We know that interferons have a pathogenic role in many rheumatic conditions, particularly lupus, interferonopathies, Sjogren's, systemic sclerosis. Studying type 1 interferon activity is really informing us about disease pathogenesis, even in COVID-19, for mortality risk and treatment response, identifying subgroups of patients. And really understanding type 1 interferons in pathways in patients may help us identify rational targets for therapy in biologically defined subgroups of patients. Now, in rheumatoid arthritis, that's type 1 interferon signature. So a interferon signature is really a pattern of gene expression that is a signal that type 1 interferon has been around. So this pattern has been identified in many rheumatic conditions, in particular lupus, systemic sclerosis, and it's identified in only a minority of rheumatoid arthritis patients. It's detectable in about 15%. But using a very highly sensitive functional assay that was developed by my primary research mentor in his early career, we were able to break new ground and detect type 1 interferon activity levels in the majority of RA patients, and it was very informative. Next slide. So in this work, we studied 2 cohorts of RA patients and measured type 1 interferon activity levels, including the alpha level and beta level, and found that pre-treatment, so a sample obtained before someone started on a TNF inhibitor, the pre-treatment type 1 interferon activity level can predict non-response to TNF inhibitor therapy in RA patients. So in our test or our initial cohort, we found that increased interferon beta associated with lack of response to TNF inhibition. So beta seems to be important itself, but we found that the ratio of beta to alpha was it seemed to be even of stronger importance. And a receiver operator curve analysis pointed at a cutoff of 1.3 as what seemed to be the optimal cutoff for the ratio. And that we then tested that ratio in a second independent RA cohort called Tetrad, a larger cohort, and found that indeed patients who had an interferon beta to alpha activity ratio above that cutoff were more likely to have non-response than good response. And again, we're aiming for good response, which is very low disease activity or remission. And in fact, no patient with that ratio achieved good response. Now, that's great. We have something to look for in blood, right? There's no commercially available, reliable way to measure type 1 interferon in patients. This is a highly sensitive functional assay. It's labor intensive and not rapidly scalable. I mean, we run it all the time. It's in a way scalable, but it's not something that is applied to the clinic. And even with the advances so far in assay development with digital PCR, we still do not have a way to measure type 1 interferon activity levels, like all of alpha and all of beta, in a reliable way. So while that is under development, which is a difficult but longer process that many of us are working on, I looked at low-hanging fruit to start to answer the question, why why does this, what is it doing? Why does it predict treatment response? Next slide. And by low-hanging fruit, I meant I looked at patients' blood. So I isolated from our rheumatoid arthritis patients pre-treatment their peripheral blood monocytes. And monocytes are known to be one of the key effector cells in rheumatoid arthritis. And I wanted to know what's happening in relation to type 1 interferon activity levels. How are they impacting these monocytes? And the quickest, easiest way to begin to look at that was to examine gene expression in patient monocytes. And we did this in fewer patients, but at a deeper level with single cell analysis. And this type of single cell analysis is different than the one you just heard about. This one was online before that technology. But one of the benefits of it, it's different in key ways, but it offers different benefits and limitations. But one of the benefits, and that was key for this study, is that it allowed me to increase the number of exceptionally rare cells in the peripheral blood so that I can study them. Whereas the other current 10x single cell model, sort of I lose in-depth, deep analysis of exceptionally rare cell types. So I was able to look at different monocyte subtypes in sufficient number. And the other is I used qPCR, which seems kind of ancient when there's RNA-seq.
Theresa, we are over time, so I would just ask you to sort of really, if you could get to the next slide so we can finish up.
I'll just really brief then. So basically what it showed was that what we found is that type 1 interferon activity is really impacting monocyte gene expression. And for the sake of time, I won't go through this plot. The colors are indicating the interferon ratio and how it's impacting gene expression in a big way. And then the second major finding is that we found sort of an on-off switch, that JAK1 expression was completely suppressed in some RA patients. And that suppression correlated strongly with the type 1 interferon activity that we had found to predict non-response. And it was completely suppressed in all their monocytes. So it was quite striking. And I think that on-off would not have been detected without this qPCR technique. So and JAK1, as Tiphanie showed in a pathway, JAK stat, they're important in pathways. And JAK1 in particular is important in the most common, the sort of what we call canonical type 1 interferon pathway. So really what this is, next click, what this is showing us is that there's differential interferon pathway activation in patients who do not respond to a TNF inhibitor. Okay, next slide. So now, and this is where my work is currently, the next two slides, and then I'm finished, is I'm examining this more directly. I was able to collaborate with Dr. Wei-King Chen in our Department of Biomedical Engineering at NYU. And we have developed a way to examine and interrogate the effects of type 1 interferon on cells in the circulation, on blood cells, and on cells in the synovial tissue. And here, I'm just explaining, I'm looking at monocultures of cells. I'm looking at effects of type 1 interferon on different cell types from patient synovial tissue cells and from patient peripheral blood monocytes in routine culture, but we lose the context. So this development of what is shown here, an organ on a chip, really is allowing us to interrogate this in a biomimetic way, but using all human cells and cells from patients. Next slide. So we've developed a perfusable synovium on a chip, or rheumatoid arthritis on a chip. And it's really a micro mimic of the joint lining of the joint itself. And it's including blood flow, which we know is part of the 3D, the in vitro structure of the joint. And so we can use patient cells, study them as they're perfused through, study them infiltrating into the joint, study effects of interferons that are put on the synovial tissue directly or in the perfusion. And our model can be further adapted to include additional cell types in the future, test additional cytokines or therapies. It has the potential to be sort of a clinical trial on a chip for using patient cells and pre-testing, their candidacy for certain treatments. This is way in the future, but this is potential, and can have a role in preclinical drug testing. And yes, this is the first vascularized device used so far as I know, is this. So I'm very excited about the potential because it allows me to really investigate what I think is important. And it also has roles to help investigate other people's work too. So other cell types of interest. So next slide. And then this is another major area, and some of the funding from the ANRF has supported the beginnings of this line of work. I'm also working with a graduate student in our Center for Data Science to really federate data from multiple sources. So my goal is to take all the information we can from a patient, clinical, biological, everything we have, and figure out what's most important and come up with a tool we can use to identify patients who should be given a certain treatment versus another. And so I'm at the beginning stages of that, really using novel techniques on the cutting edge of machine learning and artificial intelligence. Machine learning, a lot of people are doing it, but the output tends to be a black box, and we don't understand the why of how something comes together. But I'm enhancing machine learning with semantic technology. It hasn't been done in medicine yet, so I'm borrowing from the field of engineering again, but it's leading to innovation and things that are going to help us move forward. So we're really excited about it. And without the ANRF funding, I couldn't have been moving forward in these directions. Next slide. These are my collaborators, and my primary research mentor is Timothy Newald. Dr. Chen is listed here with his graduate students or in a postdoc. I also study synovial tissue from a large cohort from former Anka Katrina's group and Lars Klærsgaard, and so it's really been a global and national effort with awesome colleagues that makes translational research go forward, but without funding and support, it's impossible. So I can't thank you enough, ANRF.
All right, thank you very much, Theresa. Really interesting future directions. So we are over. I don't know, Emily, should we wrap up, or would you like us to continue with discussion?
I mean, there's been some great conversation via chat, and just want to echo the comments that are coming in, which is just so many appreciation and thanks to all of our researchers for their time today and for talking about the important work that they're doing. We we'll definitely be able to follow up with everyone who submitted a question that didn't get answered. So please feel free to continue to send those questions in or email those in. But I really just want to take the time to thank all of our presenters and our moderators today for this wonderful presentation. Thank you for your time. We're so grateful for your support, and hope you all have a great rest of your day.
Thanks.
Thanks.
Thank you very much. Bye.
Great talks, guys. All three of you. Yeah, really great. Exciting.